I don't think you'll ever be able to leave me alone with this. It's too scary. So Alex is with us. Yay. Hi, Alex. Here you are.
Hey.
Hello. Everybody else in the room saying hello to you as well. I'll get started in a minute, Alex. I just have to give the students their handouts, and then I will. So Alex, actually all of the handouts that I give the students are on should be on your Moodle site. There's one that is, like you'll probably recognize it. It looks like, get it checked the right way. Kind of looks like that in the nervous system.
All right.
Is the title. So you may be able to find that, I think you will. And there's the brain as well is on the Moodle site, and also structure of a neuron as well. So those ones I'm handing out. And then there's an electrical versus chemical transmission. And it's got acetylcholine and norepinephrine and those ones at the bottom. Now this one was on my instructor's site, so I'm not sure if you can get this one or not. Hang on a second. But if you can't get it, I'll email it to you. Okay.
Okay. So they should be under the nervous system section.
It's under the nervous system.
Okay.
So when you look at your Moodle site, it should be under the nervous system, and it's under worksheets. Should be under the worksheets. Okay. Just hand them up and then we can get started.
Okay.
How are you today? Stressed because I couldn't get in line yesterday. Oh, so many problems. Yes, we are. I said I was talking to them. If they offer this course to me, I would say yeah. If they can't manage the online stuff better, then I can't teach you because the students out there. I need it. You guys can always read. If you can get on the weekend, you can do the whole class. You don't even have to take notes off the weekend. I think that's being here. Because you're here. Well, Julie, I would rather teach through teachers here. Because you can see how they respond and you can see whether or not they're learning. And they feel like asking questions, they didn't have questions. It makes it so different. And it's as a teacher, as an instructor too, having the students respond to what you're teaching is what you need. Because you've got to know if they get it or not. It's really important. The classroom is a lot easier.
I find it's probably harder for people online to absorb and they're not really here.
Yes, and I know Alex was telling me, I asked him to just kind of tell me how it was going with the Moodle site. Not the Moodle site, but the Moodle site. Yeah. Whether he was able to hear, whether he was able to see what we've got on board and that type of thing. And he's having issues with the audio feed. Yeah. So today I'm trying to bring the microphone closer, and hopefully I'll speak up a little bit more clearly so we can see. So yeah, it's not just the teaching part, it's the whole technical part of presenting it, making sure everybody can hear. So that's what my frustration is. And I've had some frustrations, as you have, with getting on the Moodle site. So it's a little bit.
All of them. Like for me, it's everything in general online, or it's all posted on there.
Yes.
Whereas if they email it, I have access to it right there and then.
Right. Well, how about the YouTube? You probably didn't try to get the YouTube up link for the class that we had last week. So instead of having it on the Blue Jeans site, he moved it, he uploaded it onto YouTube, and then he presented it on the NBIT YouTube site. So I'm not sure if the other students who didn't attend the class have got it or not. I don't have a lot, and that's another frustration with I don't have a lot of communication. I mean, if they don't email me or I email them, I really don't hear anything about how's it going. Because I can ask you guys, well, did you understand that or how's it going? But I don't seem to get that same response from the other students that are online. So that's a little bit difficult that way. But we'll get started because this part of the course is the heaviest part. The neurological stuff is the worst. So, I mean, when we get onto the organs, it's much simpler and much easier to understand. So if you have any issues with what I've presented and you want to stop me or whatever, just stop me because it is hard to understand. I tried to go through and simplify it. You'll notice that these PowerPoints are totally different than the ones you get on your website. I've edited out a lot of the information that I thought was unnecessary. There's a lot of information on the one on the Moodle site that has to do with repolarization of nerves, and it's very complicated, and it's not something that you really, really need to know the in-depth part of it. You need to know a little bit, but not in-depth. And on the PowerPoint, the other PowerPoint, it's very in-depth, and I found it hard. So hopefully I've cleared it up a little bit for you, and it won't be so confusing.
I was going over last night on my phone.
Oh yeah.
This is stuff in the brain because I have a downloaded program like this, and it's funny, so it's just and all this stuff has been there.
I found it. So chapter 11 is neural tissue, and that's the first section. There's the other sections of the neural. Well, I'm hoping we can get through at least 2 today because it's pretty crunched, and try to get through 2. I've got the other one prepared as well, so maybe we might start the next time. Okay, so neural tissue. Great stuff. Volunteer. Here we go. It is in the anatomical divisions of the nervous system. You have the central nervous system, which is kind of like your axial skeleton, right? The middle, the central. So it's the brain and the spinal cord. So in your textbook, they refer to the brain often, well, they'll refer to cranial nerves or spinal cord as the spinal nerves. But we'll use brain and spinal cord. It's just easier to follow. So it's responsible for sensory data, and it coordinates the sensory data with motor commands. So that's what the brain and spinal cord are doing. The next division is peripheral, one here. And I'm not sure, Alex, if you can see the red. The central nervous system is the CNS, and you'll see it referred to from now on as CNS. And the peripheral nervous system is the PNS, so you'll see that. And I'm not quite sure. I have done those in red because we know what red means. Yeah, it's important. Okay. So I did them in red for that reason, but I'm hoping that online students see those red ones. So the peripheral nervous system is kind of like your appendicular skeleton, right? It's the outside part, your legs and arms and all that good stuff. So it connects the sensory receptors throughout the body, so legs and arms, and carries motor commands to the peripheral tissues and systems. And it contains what they call post-ganglionic nerve fibers. So I'll just let you write that information down. So have you read your textbook? Have you read your
Yes, that's what I
You
was
did.
doing last night.
How did you find it to read?
A lot of things, but
It's kind of overwhelming, isn't it?
it's like I read
oh my.
chapter 5.
Mhm. Yes.
One of the notes.
Yes.
And then I was going through this as I was going through it, which is like big, huge chunks of information.
Great.
And then
Right.
going through it and then stopping. And then
Mhm.
having to go back and read what I read. Like,
Great. So you can get all of it.
yeah.
That's good.
But other than that,
Well-
I think it was pretty.
Yeah. Okay. And Jill, how are you finding it? Are you reading? Are you having time to read? That's okay. No, and it's okay. We encourage you to read because there's a lot more information in there that I'm giving in the classes. But what I'm giving in the classes is what you need to pass this program. So hopefully that's going to work for you. Because I know life is busy and it's, I just don't have time. So the functional cells of the nervous system are the neurons, individual nerve cells, and the neuroglial cells. Hopefully you can read that. The supporting cells. So it's again very similar to your bone structure, right? You have a supporting system, which is your matrix in your bones, and you also have a supporting system in your nerve cells, which are these neuroglial cells. They are the most abundant cell in the nervous system, which makes a lot of sense because they're supporting everything. Now, on your handouts, I have given you a neural cell, which is the little dendrites on it. Yeah, that one. That's right. And I just drew myself a bigger one because I needed to have a reference while I have the PowerPoint stuff. I can switch back and forth. So this is my giant neuron. It's in two parts. It's in the original. Oops, no, probably can't. Anyway. So that's what I did. I just drew myself one. So it's got the dendrites, it's got the neuron cell body, it has the myofibril nodes, and this is, we're going to see this again in these PowerPoints, so don't panic about writing this all down. But that's what the neurons look like. And this here. Exactly. I love doing these because when I do them, I learn a lot. And because I'm a visual learner, and if I draw this, I remember it. If I just read it in a book, I have to read it over and over and over again to get the message. Yeah, great. So it just means more to me. Yeah. Okay. But that's what I did. So this is the other end of the neuron, where the synapses are going to be going all into that during this class. So that's your neuron. The peripheral nervous system. So we talk about peripheral, which is your outside, right? Your legs, arms, and all those good things. So it's divided into two subsystems. The efferent, which is the motor division of the peripheral nervous system. The somatic nervous system, the SNS, which you're probably not going to, you will probably not see the SNS again, but you will see somatic nervous system because it controls voluntary and involuntary reflex skeletal muscles. So it's controlling the skeletal muscles. That's an important part of that. The autonomic nervous system, the ANS, autonomic control of smooth muscle tissue, your cardiac and your glandular activity. So I'll just let you get that down. So when they talk about autonomic, it's easy to remember autonomic is automatic, right? So you have smooth muscle tissue like cardiac muscle tissue, which you do not have to think about. You are able to, your heart just keeps going whether you're paying attention or not, it's automatic. So it's easy to remember autonomic, automatic, and your heart. The somatic, of course, isn't automatic because you were telling yourself to walk and move and to do things. Right? You're giving it instructions. Joe? Yeah. A little bit longer. Okay. If I'm standing in your way, just tell me. And later on, when we're going through the PowerPoint, there will be sections where there won't be so many colors, so you won't have to worry as much. But these ones that have all the colors are the ones that you're probably going to see again. Good. So the subdivisions, they seem to split everything up into subsystems and subdivisions, which kind of makes things look. Of the peripheral nervous system are the afferent and the efferent. The sensory neurons carry the sensory information from the periphery. So if you poke your finger and it's painful, the afferent will carry that, stimulate that pain sensation to the spinal cord and to the brain. So you just have to remember that it's the afferent that carries it up to the brain. The efferent or motor neurons carry motor commands from the brain to the spinal cord to the effectors. Which one? Effectors would be the ones that are affecting your movement or what you're doing. So you poked your finger, it sent a message up to the afferent neurons. They send it off to the brain, and the efferent now is going to say, oh, well, you better pull your finger away. That's hurting. And this all happens in like a millisecond. Like it's like when you poke yourself, you pull away immediately. There's no time lapse. So this is an amazing system. Okay. So the two subdivisions of the autonomic nervous system are the sympathetic and the parasympathetic. So the sympathetic division is the fight or flight arousal, meaning if you're afraid that there's a gorilla chasing you, your sympathetic nervous system will kick in and make you run. Okay? That's what I call the fight or flight. So you're ready to fight with the gorilla or you're ready to run away from it. Personally, I'd be running away from it, so fighting with gorillas. Anyway, the parasympathetic division is rest and repose, meaning most of the time you're in the parasympathetic, right? You're not running and fighting, you're resting, you're sitting in class, you're doing things that are considered resting. So the parasympathetic is taking care of that. So now they go on to the structure of a typical neuron, which is the handout that I gave you in my picture. So I'm going to use the line because that's bigger and easier. We'll just run through it really quickly because we're going to talk about each one of them. So you have the dendrites, which are at the top I'm gonna say top of the nerve. Cell body is the nucleus. It's right here. The cell body around it, and the nucleus works as all nucleuses do in all other cells. That's the command center, right? The axon is this long, kind of rope-like thing here. On the outside of the axon are located the myelin sheath. Also on the axon are the Schwann cells, or the oligodendrocyte. The middle one here, myelin sheath, that one. And we're just gonna have a look at the picture, and then we're gonna go on and talk about each one of them. Okay. And at the very end here, you have the synaptic terminal. So if you had a nerve impulse, if you had a push finger, the message got sent up, it would be received, it would be sent down the axon out into synaptic terminal and send that message on and on through other nerves. So this is just one single neuron. So the cell bodies, we're just going to talk about each section. So the cell body, this thing with all the little branches attached to it. So the substances that are produced in the body of the nerve cell are neurotransmitters, proteins, and enzymes. The one you want to remember is the neurotransmitter. We're going to talk about that a lot more. So the cell body is responsible for transporting the substances down to the synaptic knob, which is at the other end, and that's the area right in here on my diagram. This little area down here, where they're used for nerve transmission. So the synaptic knob also transports substances back from the cell, back to the cell body from the synapse. If you had your diagram close to you, you could kind of fill in some of the blanks there too. Yeah, if you have time. Okay, good. Good. Jill, are you okay? The dendrites, those are the branches at the top here. So the projections of neurons which collect nerve impulses from other neurons, or sensory nerve endings. They're branched extensions of the cell body and they collect impulses. Now, this is probably a little bit tedious to go through each one of these, but later on we talk again about them, and we talk about nerve transmission, and you kind of have to have this in your head, what's happening. So it just gives you a kind of a micro view of a dendrite and where there's synapses around the dendrite that are sending. These are coming in from other nerves or other axons, and they are coming in and sending the message on. Just one of the link, kind of like a big chain. So the axon, which we had, is this long, skinny thing here, down.
Your colors stand out.
Oh, do they?
Yeah.
Oh, good. That one's, these yellow ones
Yeah.
are fluorescent. So that's pretty cool. Okay. So it carries electric signal, action potential. Remember action potential, that is going to come up again as we go along. So it carries an electric signal away from the cell body out to the target location. They can only travel in one way. So it starts at the top and goes out. They don't travel, electric signals don't travel backwards. They can only travel outwards. The axon can have myelinated or non-myelinated, depending on the location, size, and function. And when we talk about myelination or myelinated, we're talking about these little, or my diagram, little yellow, they're wrappers actually. They wrap around the axon. And you can either have the wrappers or not have the wrappers. The wrappers are important, but we're going to talk about that as we go on. You write fast.
Because
Well, it gives Alex time to write too, so he's probably having a hard time reading it. The axon hillock, the junction between the cell body and the axon. So that would be this area here. So this area in here. So this information here, the impulse has to build up to a certain point before it's going to be passed on. So if it builds up to a certain threshold, it will become a full-length action potential. In other words, if it doesn't build up strong enough at the axon hillock, it will not be passed on and just dissipate. You won't. If you poked your finger, but it wasn't really that painful, you know, nothing's going to be done about it. It's really very interesting because later on we'll mention how the body adjusts to pain. And for myself, I have to poke my fingers maybe about 6 times a day to do blood sugars. And I don't feel that anymore. Yeah, I don't feel it at all. I don't feel my insulin needles either. But when I'm sewing, if I poke my finger, I feel it. So my body has learned to adjust to the fact that I am poking my finger and it knows it's doing that. So it doesn't respond anymore. I don't feel it. I don't feel the pain. It's very interesting how the body has adjusted. And we'll talk. We're going to mention that later. Okay, good. The telodendra, I I have trouble pronouncing some of these words. So that's the fine extensions of the distal axon. So distal is always the furthest away, right? So that's like that fingered part here. Or it doesn't show it in mine as well, because mine is a large one. I'm just trying to find my picture. So this part, I'll get it close to the camera. I think it's working. So this part here, I'm at the wrong end. This is interesting. So you have to do it backwards. So those there are the teledendra, and that's the opposite end. So that's the distal end from the body that's over here. So each one of those little extensions, it has a synaptic terminal. And it allows one cell to connect with multiple effectors. Again, you're seeing that word, effectors. I guess you can remember by effect, it has an effect on what you're doing, like what you're responding. I can't believe how fast time is. Yeah, I know. It's like, yeah, we've been doing this for half an hour. I think, wow. Okay, good. Thank you. The synaptic terminal, and this is what my big diagram is the synaptic terminal. And that's just one of those little tips. So that's the chemical setup too, so I put all that in there. So the tip of each teledendra, where the action potential is transported to an effector, we have the effector again, site, through to either synaptic gap, the cleft, talking about that, a gap junction. Now, the synaptic gap uses chemical, and the gap junction uses electrical. And I did hand out an information sheet on the difference between electrical and chemical. So you'll have that to review. And Alex, I'll send that one to you because I kind of think it's in the instructors and you won't be able to see it. So I'll give you a hint. When you're studying for your exams, both the midterm and the final, it's heavily weighted on the nervous system. So concentrate on that. Synaptic knob, which I talked about earlier, which was this little lump at the bottom. So it's just an expanded area of the axon. It's just a little bit wider. And it contains synaptic vesicles. That's what I'm talking about. I'll talk more with you guys. I've got it written down. The reason we concentrate so much on this is the synapse is affected by medications. So if you're giving someone, the best one I can think of is it's called succinylcholine. It is a a drug that they use in anesthetics, and it actually changes how these neurotransmitters are sent across the gap. So it affects the person's breathing and it affects their consciousness. So that's why we kind of concentrate a lot on this, because it is going to affect the medications that you're giving to your patients. People with Parkinson's, all sorts of different diseases that you have to give medications for. And also, it's where all your pain control is as well. So if you're giving opiates, morphine, that type of thing, this is where it's all working at the synapse. Okay, very good. What's that? It caught me up. Good. I don't know how well you can see this. Oh, and that actually it's not coming out too badly for Alex. And the online people need something to point with. Good. Hold it. It's not bad. So as you can see here, so this is a synapse here, this little area here. It's a fluid-filled area. It's got membranes on either side. It's the presynaptic membrane is on the side closest to the neuron. And the postsynaptic is on the opposite side, but it is just the start of another neuron. Okay, it's at the other end. So you have these little vesicles. They're like little bubbles, actually, that are full of neurotransmitters. So as the neurotubules move these neurotransmitters down from the nerve body, because that's where they're created, and they move them down like a little conveyor belt, and they drop them off here. They kind of come to the edge of the membrane, they break open, and these small little neurotransmitters, which are the chemicals, come across and attach on the other side. You can see what's happening here. So any of your medications that you would be giving may work in this area, in the synaptic area, to prevent these neurotransmitters from moving across or to increase it. So that's what a lot of medications do. What else? There's also positive ions in here, and we're going to be talking about electrical impulses and chemical. And positive ions also have to be present before this synaptic event occurs. Any questions? Good. When you go home, you'll have a whole bunch of questions. Oh my God, I didn't get that.
Yeah, that's right.
Yeah, you get home and you go, oh, I don't think I get that. So the synapse is the connection between the two neurons. This is just kind of what I've gone through with you. They're the area where the neuron communicates with the next neuron. So we talked about the presynaptic neuron and the postsynaptic. So this, on my diagram, okay, hold it here. This would be, how's this doing? So this green one would be the presynaptic neuron, and the blue one would be the postsynaptic neuron on the other side. the two types of synapses, and we kind of briefly mentioned that before, was the electrical one. So it's direct physical contact between cells. And it doesn't require any chemical transmitters. And then the chemical one, which we have discussed, and I showed you those little balls of neurotransmitters, those are the chemical ones. So this word synaptic gap or synaptic cleft, you're going to see that one again too. So, I was going to say, I'm hoping somebody's running down the hall or something. I don't know. We're having a good time.
The ghosts are running around.
Like, yeah, well, it's getting close to Halloween, so there you go. Maybe we never get to talk about ghosts and stuff because Ashcroft Hospital has ghosts. It's really
I think
interesting. Yeah,
there's residents.
yes, it has ghosts too. Yeah. It's really.
It's that old, like, this is a really that old hospital, right?
Yes, that's right. Yeah.
And I was there with the doctor, and because I was thinking for the weekend, he was like, "Don't go," and I said, "Yeah, you can have one by yourself." He's like, "Don't say that, I'm here by myself."
So he was a believer too, eh?
Yeah.
Oh, it's really, some of the things that have happened are quite interesting. Let's finish up.
I thought it was funny. He didn't think it was funny.
Well, you must have been a believer then.
It was really bad at the door first year.
Is that right?
Yeah. They had somebody pass away.
Yeah. Oh, did they? Oh,
I
wow.
was living on
Yeah.
section B.
Mhm.
And we were the only, there's like 4 of us
Right.
on that level. No one on the second or third.
Right.
But we always heard somebody running around upstairs.
Is that? Oh. No. I'm a believer, so I would be going, yeah, probably. Okay. So the synaptic cleft or gap, and this is the one that I kind of showed you on here, is this area right in here, is between the two. And it does have fluid in there too to help the transmitters go across. So it's fluid filled and it separates the pre and postsynaptic membranes. The presynaptic, like we saw with the picture with the little bubbles, releases neurotransmitters, and the postsynaptic contains neurotransmitter receptors. That makes perfect sense. And in my picture here, that I have here, it is these little yellow guys down here are the receptors. And I think they just, with this particular PowerPoint, they just want to make it clear that this is a chemical synapse. So the neurotransmitters, my little balloons with the bubbles inside, these guys here, they're the chemical messengers. They're passing on the electrical impulses. They're released by the presynaptic cell, and they attach to the receptors on the postsynaptic cell. We kind of talked about that already, so you might not have to write too much. are broken down by enzymes after they've crossed, and the byproducts are reabsorbed by the synaptic knob, which is that knob at the end of my picture, and transported back to the cell body to be reused again. So it's very efficient at reusing. So just to review, because like I said, this is, if you can get your mind around this, then things are going to be much easier. So the electrical impulse is our action potential. It travels through the axon of the presynaptic cell. And once the electrical impulse reaches the synaptic terminal, the synaptic knob releases chemical transmitters. The neurotransmitter travels through the synaptic gap or cleft and is picked up by receptors in the postsynaptic cell. The neurotransmitters allow the electrical impulse or action potential to pass through the membrane to the postsynaptic cell. So that's just a quick review. Now the types of synapses, there's different types for different jobs. The neuromuscular is between, obviously muscular, between a neuron and a muscle. And is the effector as well. So it affects movement, it affects what you're doing. The neuroglandular is between a neuron and a gland. Again, affects the gland's secretion. If you were, if you were really terrified of the gorilla, it would affect glands to put out epinephrine and get you running really fast. Yeah, you and snakes. Okay. They don't really, they startle me, but I'm not really afraid of them. But I really am cautious with the rattlesnakes, right? Because we've had, one year I worked, emerged, I had 5 rattlesnake bites. Only 2 of them were envenomated, but the other ones were bites, but the snake chose not to put the venom in. But we had a lot one year. I think we had one this year. Like my girlfriend Leanne dealt with it. One of my coworkers. So yeah, I always look at the tail if I can see the tail to make sure it's not a rattlesnake.
I'm just terrified of the hole.
Yeah. Oh yeah.
That comes from like a chest and a ring, like
Somebody.
Yeah. They were like literally attacking me in the face. So then I was like, until then it was.
Right. Yeah. Exactly. Yeah. Oh, for sure. Yeah, and I know that my brother, I mean, he's 6'2", he's a big guy, and he's terrified of mice. And he's just terrified. And he said to me, and that's the same thing. At school, somebody was running around and chasing the younger kids with a mouse. Yeah. And yeah, he's got this thing about mice now. Yeah. Just see him when he had one of those little pack rats got into his truck, and it was in. And he freaked out. He had to go get his wife to chase the packer out of the truck because he was terrified. I never knew that. Okay. So functional classification of neurons. The sensory afferent and the motor efferent. We're going back to those afferent and efferent again. So the afferent delivers information from the periphery. When you poke your finger, okay, it's delivering that information. Pardon? We have talked about it, but this is a little bit more in depth. We could add, when you're doing your Cornell notes, you could kind of add it to what you've already written. Okay? But yeah, it gets a little bit more information. So the interneurons are neural cells, and they're responsible for the analysis of sensory input and the coordination of the motor outputs. So they're involved with our complex neural functions, learning, memory, planning, anything along that time, that type of work. So these interneurons are pretty important when you're studying. Better your memory. So the interneurons would work with the sensory and the motor, and as the peripheral senses come in, like the pain in your finger, the interneurons would work to tell the afferent ones to pull your finger away. I'll let you finish. These guys, I think you can see again. Okay. Types of sensory, and we're talking about the interoceptors, the exoceptors, and the proprioceptors. So interceptors, internal senses. It monitors your internal organs. Digestive, respiratory, all that. And the nerves relay sensations of taste, heat, pressure, and pain. Those are the interoceptors. Not to be confused with the interneurons, which we saw on the last page. I think this is where it gets confusing because there's so many names that are very similar. Just have to pay really close attention to the spelling. So the exoceptors are external senses. The proprioceptors monitor position and movement of the skeletal muscle. Kind of interesting that all this work is going on while I'm standing here, so I don't fall over. Cool. Yeah, it is, isn't it? The bodies are just incredible things. Yeah, it's upside down. Are you good? Yeah. Types of efferent neurons. E again, the spelling, watch the spelling on these. Somatic motor neurons that directly innervate skeletal muscles. And the visceral motor neurons, which indirectly innervate smooth muscles of the viscera. And the heart is our example again. Now later on we're going to be talking more about the sympathetic and the parasympathetic. So they synapse onto neurons located in the autonomic nervous system, the sympathetic and the parasympathetic. So they're responsible for visceral muscles and some of the glands. Other smooth muscles of the viscera would be like your digestive tract, right? Your peristalsis, that type of thing. So when we learned about muscles in our last class, you could probably remember which were the smooth muscles and which were the skeletal muscles. It all kind of builds one on the other, like each class gives more information. So this is, I think this is a little bit of a review, carries instructions from the CNS, the central nervous system, to the peripheral effectors via the effector fibers or afferents. Sorry, I don't mean to rush you. You know, there's a lot of stuff to make notes on. So the two major efferent systems, efferent are the somatic. They've got efferent over here. Like I said, you won't see those very often. Are just the CNS and the PNS. So it's to innervate skeletal muscles. Okay. Next. The autonomic is the next one. Second afferent system. The visceral motor neurons innervate other effectors, smooth muscle, cardiac muscle glands, and adipose. It's interesting you have to have nerves in your fat cells. Yes, you couldn't lay them down without the nerves to tell the body what to do. So, yeah, okay, I get why they have it. Okay. So this is just a diagram. I think that one's available on Moodle. I don't know for sure. Or you could just look at the PowerPoint maybe and pull it off of there. So it just gives you the breakdown of the central nervous system and the peripheral nervous system, the afferent division, the efferent division, and then somatic and visceral. That's a lot to remember, isn't it? Remember, like you don't have to go remember all of these. This is more of a, okay, what does it do? But you will have to remember the divisions, like these top. We're going to talk about the neuroglia, which we had talked, I was earlier on in the PowerPoint. The neuroglia are the ones that are the most abundant cells, right? They are the support and protection. They are equivalent, like I said earlier, to the matrix of your bones. So they're supporting all the system. What? It's your birthday? Oh, tomorrow. Well, happy birthday tomorrow. Well, that's awesome. Is she like 18 or something? Yeah. Really? Oh my gosh, you look like you're like 16. Oh, that's good. Oh, Are you doing anything special?
Yeah, my boyfriend and I are going down to Vancouver for my
Oh.
game.
Oh, great. Good. Excellent. Celebrate. Yeah, I'm all for celebrating the good stuff because, you know, it seems that people can always show up for funerals, but they can't show up for graduations. They can't show up for all the other things that we celebrate. So it's like, I would rather go to graduation. So the types of neuroglial cells in the CNS, that gives you different types, and they're all listed here. We've already seen this oligodendrocytes in our picture that we had earlier. The astrocytes is kind of interesting because astro means star or astrology or astro. And these are shaped like stars. They have long fingers. They spread. Astro. Yeah, it's like a star. I think I have, if I can find under all this, I have a, unfortunately, they're kind of, I'll try to hold it up for the people that are online, then I'll hand it back to you. Let's see if we can get this up here. So, oops, this is rather fun to do this. We can see the different types, their shapes of cells. Okay. And I'll just hand them around here. Actually, my mother died from a brain tumor. She had an astrocytoma, meaning it was in these astrocytes. And the problem with the astrocytomas, with cancers, is that because they're star-shaped and they have all these fingers, yeah, they spread. So you can't go in and surgically take them out. They can only get the center portion and the little fingers have spread. So having astrocytomas are not good for cancer patients. Terrible. Yeah. So like they took out the center, what they did with my mom, they took out the center core. They had all those little points that they couldn't get to, and so she had to go for radiation and chemo, try to get all those little points of that star. She did extremely well. Amazing lady. She had prognosis was 2 weeks to 2 months, and she lived for 10 years.
Wow.
And the last 2 years were not a good quality of life, but she had 8 years of really quite a good quality of life. So it was worth her going through all that treatment.
Yeah.
So that was great. Okay, I'm just trying to think what else I was going to tell you here. On we go. So it just gives you a little bit more information about these cells. The ependymal cells, they're in the spinal column, central canal, and the ventricles of the brain. They form a lining in these areas, similar to an epithelium, right? So we're back to the skin again, right? Yeah. So it's similar to that, they're a lining. They have little cilia. When they talk about cilia, they mean little fingers, like little hairs, little fingers. Right? And that help to circulate the cerebrospinal fluid through the central nervous system. And this is an important part too. These cells secrete cerebral spinal fluid. And I'm sure you guys know what cerebral spinal fluid is. It's just the fluid that circulates around your brain, down your spinal column, and back up again. And it's a cushion. Keeps the brain safe. The astrocytes, which I told you that my mom had a brain tumor, which was made of astrocytes. It's the most large and abundant glial cell, and it maintains the blood-brain barrier. Now, the blood-brain barrier prevents infections from going through into the neural tissue. There are some different types of chemicals and different diseases that will cross the blood-brain barrier. I believe that AIDS is one of them. I'm not really sure. But there are lots of different chemicals that will cross it. But it's a protection for the brain. The astrocytes create a semi-rigid skeleton that holds the neurons together within the central nervous system. They also stabilize damaged neural tissue, and they help the neural tissue to regenerate as well. The astrocytes are also responsible for development during the fetal development. They guide the neurons during fetal development. And they control the environment as well within the nervous system. Pull your hair out. I've had enough of this. I'm pulling my hair out. Yeah, I felt like this when I was reading. I thought, oh my good grief, there's a lot of stuff here. I'm supposed to filter it all out and make it low and make it simple.
That was me last night reading.
Trying to filter it.
Yeah.
Yeah.
I was so exhausted after I was trying to go through it. And I just like, as soon as I closed it, it was like not even minutes after.
Yeah. I used to do that in nurses training. I used to read my, any of my nursing books, right? Anatomy or anything, and go to sleep immediately. Like it's the best sleep aid ever. Yeah, I know. Okay. The astrocytes control the interstitial environment by regulating sodium, potassium, carbon dioxide. They transport nutrients between the filaries. And because this is all in white, we know we don't have to write it all down. Okay. But they do have a job with controlling the control of the vessel size and volume of blood, absorbing and recycling the neurotransmitters, and releasing chemicals that either enhance or restrict communication across the synaptic membrane. So they're responsible for the controlled environment, right? And that's what you have to remember. They're responsible for the environment. The oligodendrites or dendrocytes, I should say, have extensions that wrap around the axons to form the myelin sheath. Now, the myelin sheath was in this picture, which wraps around, flopping around, which wraps around the axon. Okay. So the myelin sheath is responsible for improving the impulses. So it kind of wraps around and becomes an insulator, and it actually improves the impulses that go through. It makes them stronger, it makes them last longer. Now the little gaps that are between each little piece of myelin sheath, like these little gaps in here, they're called the node of Ranvier. And that's just this little gap in here. Because the nerve impulse actually can go through and it can actually jump those gaps. There are gaps between the little myelin sheaths, but the nerve impulses can jump them. So they're not connected. So some of the nerves actually have a myelin sheath and some don't, depending on what their purpose is. Now, the myelin sheath is what's affected in multiple sclerosis, MS. Yeah, okay, because the myelin sheath itself is either damaged and then it scars and then inflammation starts and then the person has difficulty with speech, with sight, any area that's affected, the myelin has been affected, they start having issues with walking, posture, that type of thing, because that myelin sheath has been damaged. So white matter and gray matter. Ta-da, new subject. It's about 2 o'clock. Do you want to have a break? And then we'll start with this new thing? Okay. Okay, so we'll take a break. 20 minutes or... So how about if we come back at 10 after? Would that be good? Yeah. Okay. Great. So Alex, we're going to take a break till 10 after.
Sounds good.
Thank you. If they don't get it back. Oh, let's see.
Just passed. It's next week, if I don't pass the oncology, then but all in all, for myself, if I'll write my desk, then it won't take it out. Yeah. Like I'm here. You like the map? The map.
what I found is like, you don't know about this. In my head, it was like it's easier to like give myself less privilege than it is to push myself. Like, you know, you got this. And it's just reading and being repetitive and doing this. Yeah. Put your phones up on the line. Because she said that there's too much cell phone usage and they actually had a concert for it. So I was just wondering what else. Yeah. I think so, and then I think what she's from the point where she's not frustrated because she feels like she's not being listened to. I think that's where some started side. And like I don't think that she approached it the right way. Like she just like literally like right on Nancy and then she was like, whoa, like where did that come from? Like I really didn't know how. Well, and that will work. Well, that's what she said. She said if that's how it's going to be, she'll have her phone and she'll know your kid's name on there. And she said if it rings for your kid, then she'll tell them. But other than that, then that's. So that's what she said, that if we can't control that, then she just going to control. If I hadn't done that,
That's a constant problem. I had students in another class with the same problem, that they were working as a security guard at the hospital, and they would phone her all the time for work, and she was just casual. So it became that, yeah, okay, you can have yours, but everybody else has to turn in theirs, and that wasn't fair. So it gets to be a big issue, the cell phone
I think
access.
that's what it was, it's just because of the focus on the class.
Mhm.
Talking with finding it.
Yeah,
Looking at her, and was they were looking at her.
great.
And
Yes.
then she noticed like right off in the
Mhm.
class, she said, therapy, writing down information, all of what we're doing.
Yeah.
I was like, wow, but she kind of came off really mean about it.
Right. Oh,
I think she was just frustrated.
I think too that, I mean, it's a whole new era with technology, right? I mean,
It is.
you didn't have that in your classrooms before. So this is the thing. And I know, I mean, I got rid of my landline, so now I have just the cell phone. And yeah, it's like if you lose it or you can't find it, it's like, I can't find my phone. I can't manage now because I can't find my phone.
Like, it's like that now too, where like I can get it to you phone now, it's like, this phone, he's like all over the place, like, where did I put it, where did I do it?
Exactly. So we're just going to go on to different, we're still dealing with the nerve cells, but we're going on to the, in the brain. So the white matter and gray matter in the brain. The white matter reaches the CNS with many myelinated nerves. So they have the covering, right? The myelin covering on them. So that's what the white matter is. And the gray matter is opposite, which is unmyelinated. And they're all within the central nervous system. We're not talking about the peripheral right now, just the central. Pardon me? Maybe eyes are getting better. Good. Somebody had onions? Yeah. What did you have? Salad?
Yeah.
Oh. No, that's okay. I don't care. Something one smell that really bothers me, and that's the tar that they put on the roads. I can't, I just like lose my breath and I can't even breathe when I drive by and they're paving. Are we good? Yep. Okay. So now they talk about the microglia, which are the smallest. Well, micro means smallest, obviously. And there's Daniel's favorite word again. Psychocytes. They migrate to the neural system and they're the janitors, right? They're cleaning things up. They're getting rid of bacteria and viruses and anything that is a waste product. ganglia. I don't know if you had people or know of people who have a ganglia. They say they have a ganglia. It's in their arm or something like that. And it's a little lump. And you can actually see it. And yeah, people have mentioned that. Yeah, that's right.
I had it cut out, but it was like,
Yeah.
it was the whole way, and it was like a big lump.
Right.
It was like, it wasn't like doing anything.
No, it
was
doesn't
just the
hurt.
fact that It was there.
Yeah.
And it was like getting, to me, it was getting bigger. And it was like, he's like, can you live with that? I'm like, well, that's gross. Like, no, I don't want to live with that.
Yeah. Right. And I was young as a teenager. I had one on the inside of my arm here. And my mom took me to the doctor, and he said, "Oh, just hit it with a book. It'll go away." Yeah. And actually, we did. We smacked it, and it kind of dissipated. It was quite unusual. Anyways, because we've talked about the lumps, it is clumps of cell bodies and dendrites that form intersection. The peripheral nervous system and the central nervous system. And they're the relay point. So I don't know why they would grow out of control like that, but I obviously, yeah. And as far as I know, they don't cause any pain or any issues at all. So they're surrounded by the ganglia are surrounded by the central nervous system neuroglial cells, satellite cells, or Schwann cells. Those are just different names for that. I've only ever seen the satellite cell come up in the sentence, and it wasn't really related, so I don't. You'll see it, but it's not really important to learn. Okay. Good. Neuroglial cells of the peripheral nervous system. So the satellite cells and the Schwann cells we're talking about. They regulate the environment again around the neurons. So the Schwann cells more are the protective sheath, and when I had my big picture up here, the Schwann cell was part of the myelin sheath. It's like it's the sheath that grows around the portion of the axon from the myelin sheath, so it grows around. So it's just to protect. Neural response to injury. This is interesting to the fact that the peripheral nerves can regenerate. So, I mean, if you have, say you cut your finger off and the doctor reattaches your finger, they can reattach the nerves as well, and they will regenerate. Yeah. Mm-hmm. Great. Yeah.
They don't look normal anymore.
But they're functional. Yeah. They have nerve impulses to the fingertips. Yeah.
There was a self-esteem thing for me. It's like, yeah, I'm really crazy, but then I'm lucky to have them.
Yes. Yeah, that's true. Yeah, you need your fingers. Yeah. So again, they talk about the length of pressure. And we talked about the compartment syndrome last time, remember? Do you know about the compartment syndrome? Because that's what you missed last time, so I might just go through it again. So, so the length of pressure on the nerve, resolving lack of oxygen and nutrients. And we talked early when we first started, we talked about what cells need: oxygen, nutrients, and water, right? So they need all those things. So the compartment syndrome, we talked about it last time, and I have got a bunch of stuff written on the board, so, but I'll explain it briefly. It's in the muscle tissue, because this was from the muscle portion last time that you missed. The muscle itself has sheaths on it, right? It's all sorts of sheaths, one on top of the other. And if the muscle becomes damaged, like in a torn ligament, a fracture, any sort of, quite severe damage, inside the sheath, these compartments may fill up with blood, and the pressure from the blood and the fluids from the damage presses on the outside, and that what happens is a compartment syndrome, meaning the nerves lack innervation. There's no oxygen, no nutrients to the cells. So what happens is that muscle tissue may die. But this is a similar sort of thing, the pressure on the nerve, where the nerve will die, and that would be going along with compartment syndrome. Because if you have pressure on the nerve, you're going to lose your nerve innervation. And we talked about color, warmth, sensation, and movement. With compartment syndrome, that's something you're watching. You would be watching for the color of the limb, the cap refill, and of course sensation. So if the limb is, if you had a fracture in your lower arm here, and you had a cast on it, because the cast is holding everything nice and tight, makes the compartment syndrome worse. You may lose sensation. You'll get numbness and tingling in your hands or your foot, depending on your distal portion to where the injury was. That's something that you're going to see in nursing quite a bit and be watching for, is compartment syndrome. Is that okay, Danielle? That's a brief explanation of what it is.
Like if I was on compartment syndrome,
Mm-hmm.
just think suffocation.
Yeah, of the cells, and it's from the damage. And usually nurses and doctors make it worse sometimes. If we put a cast on a leg or a limb and we hold it in even tighter, then there's more damage done. So that's oftentimes you will see people come in and all they'll do with a fracture is to wait for the swelling to go down. They'll just put a back slab on it and then put a tensor on it. And that's just to prevent compartment syndrome from happening. So that's part of the reason they do it that way. But it is just from the swelling. So peripheral nerve regeneration, it begins with Walleran degeneration. So the axon distal to the injury degenerates, the macrophages migrate and clean up the debris, and the swan cells do not degenerate. So the swan cells form a cord that acts as a pathway for new growth along the original pathway, and the new axon grows in. So the swan cells are the wrap the new axon. So it's interesting that it begins with the nerve cell or the degenerating, and then it is cleaned up, and then the swan cells kind of wrap themselves around and regrow a new axon. Kind of cool. So nerve regeneration in the central nervous system is very limited. So when you're talking about people with spinal cord injuries, because the spinal cord is central, right, central nervous system, and you have, well, Rick Hansen is a perfect example with spinal cord injury, that has never regenerated. And the scarring also creates more problems too. So just read through this. So many axons are involved when the damage occurs because the CNS neurons are tightly packed. The astrocytes, again, produce a scar tissue preventing reconnection. Astrocytes release chemicals that block axon growth. So you'll see this type of thing with seizure disorders. Right? If you have Seizure disorders often form scar tissue, and that's where they misfire from that scar tissue, and then the person may have seizures. 5 membrane pulses involving ion movement, electrical signals. This is the hard one. That's the one that's on behind the board here. I think we should go through it first on the board or after. Go through it first. Turn this off. So the axon, the main nerve fiber of the neuron, which is this guy here, this long skinny guy, of the body. So I've drawn myself an axon fiber here. Okay. On the outside are sodium ions, which are positively charged sodium ions. On the inside, there are negative ions. So this membrane is in between the positive and negative. When the axon is resting, that means it's not sending along a nerve signal. All the positive ions are on the outside. So they're positive sodium ions. And as the electrical impulse, or as the impulse goes through, they switch sides. Right? So this is the impulse here, where the positive ions start to move to the center or inside of the neuron, the axon. And they call this depolarization. So it depolarizes. And then as that stimulus moves on, because it'll move on all the way down here like a wave, it repolarizes on the other side of the stimuli. So you poke your finger, and that message is being sent along to the brain. I hurt my finger, and as this message is sent along, this depolarizing and repolarizing occurs. So there's only really 3 phases that you have to remember. The resting, which is all of the positive ions on the outside and all of the negative ions on the inside. The depolarizing, which is the positive ions moving into the inside and the negatives going to the outside, and then the repolarizing, which means repolarizing means just returning to resting state. So repolarizing is returning to the resting state. You can remember from the RE at the front of those. So the 2 ions that are involved in this are potassium, K positive, right, and sodium, Na positive as well. They're both positive ions. So the potassium works with the sodium, which is in the highest concentration, to depolarize and repolarize neurons. The resting membrane potential is determined by potassium, meaning potassium is more involved in the resting part of this stimuli. So did that make everything all blurry and fuzzy and don't know what's going on? Because I thought I'd go through it first because it's going to go through it on the PowerPoint. And you'll see this again. This type of thing. I don't know how this came up. Okay. So nerve impulses, all they are waves of electrical stimuli that travel along the plasma membrane. And it can be anything from poking your finger to a temperature change. You pick up a hot pan or something like that. Pressure, when you go to lift something, you have to put a lot of pressure on it to lift, that type of thing. That would be an electrical, that would be a stimuli. So again, to repeat what I just told you, the resting neuron has a positive charge on the outside and a negative charge on the inside, and that's called polarization. So the positive charge is in the form of an excess of sodium ions on the outside of the membrane. Good? Okay. No, there's no rush. I didn't do a review this morning because I knew how much content we had. So I better not review anything. But the review was the compartment syndrome. We did that. So that wasn't so bad. Okay. So depolarization. So the sodium channel is open and the sodium goes into the cell. The inside of the membrane becomes positive, the outside becomes negative. And I just put this down here as your sodium